G'day everyone and welcome to this Share Cafe Due Diligence webinar. Today we're taking a deep dive into Antisense Therapeutics, ASX code ANP, market cap of $135 million with a one-year return of up 105%. Now in a first today we have three directors presenting. This really is institutional access and we're proud to keep developing our webinars in order to provide more insight into listed companies. Today, we're going to start with a brief overview of Antisense with Managing Director Mark Diamond, and then we're going to take a deep dive with directors Dr. Charmaine Gittleson and Dr. Gill Price as they discuss Duchenne's, the phase 2 trial, clinical development plans, and then we'll discuss the competitive landscape, market potential, and commercialization considerations. So at the end of each section, we'll stop, we'll allow time for questions. So please type your questions in the box provided. So let's make a start. I'll hand over to Managing Director Mark Diamond, who will introduce Antisense Therapeutics. Thanks, Mark.
Oh, thanks, Tim. Firstly, it is a pleasure to be presenting on Antisense Therapeutics today. It's also very pleasing to be able to have on the call today two of our new directors, Charmaine and Gill. So, again, thank you very much to Share Cafe for this opportunity to overview the company. Next slide, thanks. So we will be, as part of the presentation, making forward-looking statements. So they should be treated as such as at-risk statements. I'll go to the next slide, thanks. So it is very pleasing to be presenting today, not only to be introducing our new directors to shareholders, but also because it follows two very significant events for the company. The first was that we recently reported a major advancement in the development of our lead program for Duchenne muscular dystrophy. That is that we have seen from the European Medicines Agency a positive opinion on our phase 2b/3 clinical trial that we're looking to run in Europe. So this essentially positions us as one study away from being able to take our drug to market in the second largest pharmaceutical market in the world. It also puts us in very rare company within the Australian listed biotechnology context, because there's only a handful of companies, listed companies, that have programs in this advanced or final stage of clinical development. And the majority of those have market capitalizations, many multiples beyond where Antisense Therapeutics is currently valued. So we obviously see this as being a tremendous value creation opportunity for the company as we launch into this last stage of clinical development. The other very significant news, of course, is that that we did in concert, announce in concert with this major milestone from a development perspective, a successful capital raising where we were able to place $20 million with institutional and sophisticated investors. This has substantially strengthened the balance sheet of the company. In fact, it's had us accessing the most capital in the history of the company. So it really positions us to be able to accelerate our plans to advance this program in Europe, and also to be able to move on other initiatives, including moving AT1102 forward in other disease settings. So again, a really exciting time for the company to be updating on our progress. Next slide, thank you. So this slide overviews the share price performance of the company. Since we started the program in Duchenne muscular dystrophy, when we raised capital at 2.4 cents, when the company had a market capitalization of 5 million, until the most recent financing, which we did at 24 cents, and with the market cap today at over 130 million, as Tim highlighted. The major shareholder of the company listed here is Platinum Asset Management. They are, for those unfamiliar with them, they're an Australian-based investment fund with over $22 billion under management. And Platinum Asset management have been an investor with the company, supported the company since we started the program in Duchenne muscular dystrophy. So they've participated in all the capital raisings that we've done since we have initiated this clinical program in Duchenne muscular dystrophy. And you see here on the slide that every time we've been back to the market, we've been able to raise capital at higher prices. And we're really very, very pleased to have had that ongoing support from Platinum Asset Management, but also to be able to report that we've been able to raise capital to get us to a particular value inflection point, and then to be able to increase the value of the company in the process. I think, along with the solid progress we've made from a development perspective, impacting the positive share price performance, I think another key feature has been the analyst coverage that is backing the company. We have listed here, you'll see 5 of the local biotech analysts that have positive recommendations on the company. So that research is available on the company's website as well too. Next slide, thank you. So the company has a very experienced and internationally recognised board of directors. Time doesn't permit to go through all the members of the board, but would just highlight the recent appointments of Charmaine and Gill. Both come with very deep both clinical development and commercialization experience. The appointment of Gill and Charmaine has been timed with this move of the company into this final stage of clinical development. So it has been the intent of the board to strengthen its experience in clinical development and to prepare for this commercialization of the drug in Duchenne muscular dystrophy. So I'm delighted that both Gill and Charmaine are available today to present on the company, and I think shareholders should be excited about the opportunity to hear of their views and experiences around the company. So I'll hand over, Tim, to any questions before having Charmaine talk to the development plans.
Thanks, Mark. I think there's one more slide there on the management team, but we might move that forward in the interest of time. Can you talk about, obviously you raised $20 million successfully with institutions like Platinum. They actually have their own international healthcare fund, but there's a retail entitlement to come. There's lots of chatter around that. That's at 24 cents. Is there a strategy around that if retail doesn't take up their full entitlement?
Yeah, thanks, Tim. So the capital raising that we announced did provide a funding path all the way through to trial results, and a feature there, a part of that was the entitlement offer that you referred to. What we were keen to do was to allow investors, our retail investors, to come in on the same terms effectively as those that participate in the placement. So if with the entitlement offer, if the entitlement offer is not all taken up, then we have the opportunity to subsequently place any shortfall, have 3 months to be able to place the shortfall. The reality is, Tim, the company is in need of having those funds in place today to allow us to advance the program forward, to be able to look to set up the phase 2b/3 clinical trial in Europe. We've got plenty of cash to allow us to submit our trial applications and to get those through to approval to a point where we could look to be able to initiate the study. So we're anticipating being able to initiate the study around the middle of next year. So essentially, we've got all that time to be able to bring in any additional funds that we may require to be able to move to the next phase of development, which would be to the reporting of a futility analysis that we have planned as part of the study.
Okay, thank you, Mark. We'll now introduce Dr. Charmaine Gittleson, and she'll discuss ATL1102 Duchenne's and the phase 2 trial. Over to you, please, Charmaine.
Okay, well, Thank you very much. Thanks for having me, and good to be here and to be speaking to you. Today, I'm going to talk about antisense oligonucleotide drugs, how they work, and why for Duchenne's. Now, I know many of you who have followed the company for some time will be aware of this, but we have many new shareholders, and I want to take this opportunity to explain this to them. So the organization is built on the platform, the technology platform of these drugs called antisense oligonucleotide drugs. They are, thank you, they are very targeted, novel therapies that can impact how an individual cell works. If you look at the graphic on the right-hand side, this is right deep within the cell, DNA that is transferred from its genetic code ultimately to a protein or an enzyme showing on the right-hand side that is then able to exert an effect. People know about gene therapy, that would be targeting where you see the blue DNA. There's now things that target messenger RNA, such as the COVID vaccine, where you actually put messenger RNA into people. And there are now also these antisense drugs, which are even more targeted, which can change the protein that a DNA messenger RNA manufactures. The proteins that you can make doing this, you can either make protein that the body already makes and hope that that helps the individual, or you can look at proteins that are causing problems and you can change those proteins and make them non-functional so they can no longer cause disease. And that is what Antisense has taken the focus on, is on the type of antisense drug that changes the proteins that are made so that they are no longer causing a negative effect in the body that one doesn't want. This type of technology was first brought to the fore by Ionis Pharmaceuticals. They are the world leaders in this area, and Antisense has been partnered with them. If you move to the next slide, please. So if you have a look at an area where a protein causes problems, inflammation is one of the models that represents this the best. And what happens when you have chronic inflammation, particularly in muscle tissue, as happens in the disease that we're going to discuss today, unfortunately, the abnormal muscle sets up an inflammatory response. It attracts to it cells of inflammation that get there and try and fix the problem and actually, unfortunately, cause more damage. And when you get this chronic damage occurring in a muscle, the muscle tries to repair and it remodels and it lays down abnormal fibrous tissue or collagen and becomes very stiff and unable to function. And it's done or driven in part by these immune cells, or T cells, specifically called CD49 T cells, that go from the bloodstream. They're normal T cells. And you want them normally when you say have cut yourself and you have a bit of inflammation and you want to clear that, and it does, and it goes away. But unfortunately, when you've got a chronic problem, as in Duchenne's, these T cells move into the blood, from the blood into the muscle, and they set up this ongoing phase of inflammation that the body has to repair, which it does by this fibrous tissue. Now, what's been shown over time is that if you look at mice disease models and then take biopsies from patients with this chronic inflammatory changes, that their tissue actually shows these very, very high levels of these CD49 T cells. We call them CD49 specifically. They're a subset of T cells, of immune cells, because they have on their surface a certain protein that can interact with other tissue that allows them to move into the other tissue. And that little factor on the surface is called an integrin, and it's named CD49. What we've been able to demonstrate over time in both animal studies, and I'll come to the human part, is that the drug that Antisense has, ATL1102, is able to change this picture by if I go back to the previous, I don't want to go back to the previous slide, but just make you think back, what it does is it actually changes the way that CD49 protein is made and what it looks like. So it's still there, but it doesn't function properly, and therefore these T cells can't migrate out into the muscle tissue. If we move on to the next slide, please. So Duchenne's muscular dystrophy is a primary target for this type of approach, because when these patients have their condition, which we'll talk about in a little while, they set up this chronic inflammation in the muscle. Another condition that occurs is multiple sclerosis, and there are some other also in chronic inflammatory muscle diseases where this occurs. And there's now been a lot of work done where you're able to take biopsies from these patients and mix it with drugs that alter the effect or function of the CD49, and you can show that you can decrease a lot of the abnormal damaging processes that occur. Move to the next slide. A lot of this has come from research collaborations, and Antisense has had research collaborations over the years that have demonstrated this basic research data. What one usually does in development, and certainly in my experience, is that before you come into a clinical trial and simply give patients a drug, it's best to choose the optimal drug and the optimal way to give it. And you also want to demonstrate that it's safe, but you want to understand the mechanism of action. And Antisense is partnered at the moment with the Murdoch Research Institute, and we're doing a lot of work having a look at this in-depth mechanism of action, not only to understand which patients with Duchenne's would best benefit from the mechanism, but we're also starting to have a look at what other muscle diseases might one be able to apply this approach to be able to treat this fibrosis and prevent the damage and ongoing damage in patients. I just want to stress that this takes a lot of time and a lot of expertise. You have to set up these experiments. You have to work out which models are going to be able to work. There's a lot of collaboration that occurs across researchers across the world. If I move to the next slide, please. The data that comes out of it, and I promise I'm not going to speak to this complex data, but the data that comes out of it is really complex. And in order for us to have a look and say we're going to believe in it, we often run these experiments more than once and in more than one model. So at the moment, we have, out of a large amount of potential data points, we have a set of data that has really given us confidence into the mechanism of action, which makes us believe that this approach of ameliorating this fibrosis and the movement of the CD49 T cells into muscle tissue is going to help the patients who are unable to walk with DMD because their disease has gone so far, as well as those who are earlier on in the disease process in whom the fibrosis is starting. And I'm going to explain to you in a moment how these two parts of DMD. Suffice to say that this is really promising. We are now able to, based on this information, start to have a look at other muscular diseases and see whether we can influence the regeneration. There's a lot of work to be done, thousands of data points to go through, plus a lot of other experiments before we feel we're in a position to say that if we took the drug and put it into other patients with other diseases, we would feel confident that they're potentially going to get a benefit. But right now where we are is able to focus on Duchenne's muscular dystrophy. So what is it? Let's go on to the next slide, please. Duchenne's is a very sad illness, and it's a genetic disease that a patient can inherit and can be also from a mutation that occurs within that infant, within the fetus while it's developing. It's very sad because the child, the little boy, is born absolutely normally, bringing joy to his family, and then the families start to notice that there are delays in the child's progress and motor milestones. It's progressive. It results in muscles, particularly the large muscles, the muscles of the leg and the arm, becoming completely dysfunctional. So the boy becomes wheelchair bound, at which stage they're called non-ambulant. Up until then, they're ambulant and they can walk, but they are weak, unable to get up, weakness in the arm. So the things that you would expect a little boy to be able to do, run and play, catch a ball, feed himself, learn to brush his teeth, learn to put his clothes on, these children are unable to do. And by the time they get to their late teens, 20s, it starts to affect the muscles of the chest wall that allow you to breathe, that expand your lungs and pull air in. And as soon as that happens, and these young people are no longer able to breathe, they end up on ventilators. And unfortunately, there's 100% mortality and these patients die. There has been very little in the way of being able to help these patients. There are companies that are trying genetic approaches to try and give them gene therapy. However, when you give gene therapy, you have to target the very specific mutation that that child has. It's not something that you can then give to all DMD children. The same as some of the current therapies that are known as exon skipping, that try to skip the part of the mRNA that is coding for the abnormality in the protein. You have to be very specific and targeted almost in a personalized way to the abnormality that has. There has been nothing other than corticosteroids that can treat the inflammation and the damage that occurs in the muscle that is progressive. And steroids are really an awful drug to be on for a long period of time. They are fraught with side effects. And in other disease areas where steroids have been used, for example, asthma, that initially was reliant on chronic corticosteroids, research in other diseases have all worked to find drugs that are corticosteroid sparing. Doctors really, we do not like to use corticosteroids on an ongoing basis. And for young children, they stunt growth, etc. So what we're looking to do is bring a product that can be used in all people who have DMD, and also that would have less side effects or minimal side effects, and would be able to stop this awful progression of the ongoing weakness of the muscle due to the damage that is occurring. If we can go to the next slide, please. So as I said, affects male births, and a number of the boys, well, when they start, they're diagnosed, they usually are ambulant. They're diagnosed with weakness and this delay in milestones. But unfortunately, what one has to tell the parents or the caregivers of these boys is that your son is going to progress, in the majority of cases, to being non-ambulant or wheelchair bound. And even if the boy is able to stand and walk a little bit, they're still going to spend most of their day in a wheelchair and then get progressive weakness of the upper body. Can I move to the next slide, please? To really give you the best idea of what this is and how devastating this is, we thought it best to show you a DVD. And this has been put together by the Duchenne's Foundation, Save Our Sons, who are really committed to trying to find ways to help their patients. So please, Tim, if you could play this video for us.
In the moment between your first and second heartbeat, I finally understood time. I felt its forward march in a swirl of blood between our bodies, in the ripple of our futures unfolding and intertwining. Time split your face into its first smile, lifted your wobbly legs into first steps. But I was never ready for it to change the you I already loved. Time, leave him as he is, I begged. He is perfect now. And now. And now. But time took you anyway. And now I wait, dreading the space between this heartbeat and the last.
Thank you. So let's move to the next slide, please. So why Duchenne's muscular dystrophy? I think we've covered this to explain that what's happening in these boys is the abnormality in the gene causing this abnormal protein dystrophin, and this sets up the inflammation. But unfortunately, it progresses the inflammation and damages muscles. And so really, what this disease is crying out for is this huge unmet need to be able to address this ongoing inflammation and damage to the muscles. Now, the parents, the boys, the families would like something yesterday to be able to treat these patients. Us as clinicians, we're also desperate to be able to find something and be able to move something through the clinic as quickly as possible. And we do appreciate that as investors, you also want us to be moving as quickly as possible, not only for these patients, but also because, let's face it, there's a return on the investment. But, you know, when you're dealing with rare diseases and patients who already have much going on in their lives, you have to be so careful when you give them a new drug that you are not going to cause more unforeseen damage as an unintended consequence. And so you will see that at Antisense, they have stepped, or now we, with myself and Gill on board, we are stepping through this in a very responsible manner. The work has been done at a preclinical level, the scientific level, and that scientific work at the bench is ongoing. If we can move to the next slide, please. We have taken the product and put it into a small number of boys to test the hypothesis that if we gave this to wheelchair-bound, non-ambulant boys, that one, we would not cause more damage, that we could do it on an ongoing basis. So it's been trialed for 24 weeks, and that we could have some positive impact on their disease progression. What you expect in boys where there's really nothing now for them but steroids, is that they will maybe stay stable in their disease, but unfortunately, even with the steroids, they deteriorate over time. So when this study was done, really what Professor Voigt wanted to see was, could you get boys to just be stable, maybe be lucky to have a few boys improve, but at least try and get them stable of where they are. That is a huge win for patients and for their families. So the study was done. I'll move to the next slide, which is my last slide. And what was really pleasing to see, even though it was in a small number, that there were a significant number of boys that were able to keep their strength in the upper arm, which is measured by what is called the pull-2. It's now becoming the global standard of how you measure strength for these boys. Remember, they can't walk. Their legs don't work. You really can't do much with them. Many of them can't take a hairbrush to brush their hair, but you want to try and understand what strength they might have in the upper limb. What was seen over the 6-month period was that a number of the boys actually remained static in terms of their pull-2. They did not deteriorate. And compare that to boys that are in what's known as a natural history cohort that are not getting an experimental drug that would deteriorate over that time. So that was really a positive outcome for this program, that it was safe, that you could give it and you could start to see some impact on them. So the next step in this is to now take it to a phase 3 study that is what we call phase 3 or phase 2B. That is the last study that one does, where you collect larger information, you compare it against other groups within your clinical trial, and Gill is going to take you through that in a moment, and that allows you to take that to the regulators and show them with convincing evidence that you have something that is of benefit, and you can register your product. So we've gone through the process. We're now at a very exciting phase. And before Gill takes you through that, I'll hand back to Tim in case there are some questions.
Thank you, Charmaine. It's a very powerful video, and it's you're doing some significant and important work. And I think that's a very good reminder to everyone, including shareholders. Now, can you talk us through what regarding the phase 2 trial results do you regard as the most compelling?
I think the pull to, that is what everybody has been really excited about. The fact that these boys did not deteriorate. And when you have a look at other, so that's really exciting. Because that's what parents are dealing with. They're dealing with the child in front of them, seeing this deterioration in strength. And so when you see that deterioration slowing down or not occurring, that is really exciting for a mom to know that whatever her son is able to do whilst on this drug, he's going to continue being able to do that. If you take it to a more scientific level, then what we really were excited to see as well was that, the things that we're looking for, that the CD49 lymphocytes, this integrin and the changes there, as well as when we did the more specialized testing with MRI, looking at what was happening in the muscles, that everything that we want to see occurring when you give this drug, everything was going in the same direction and in the right direction. So you start to know, well, something's happening. It doesn't happen, by magic.
And there's a question here, can Charmaine address the outlier patient results?
So these are really challenging to understand. And what we know from other studies that have been done, in fact, it's a study that if you go back and have a look afterwards, you'll see it's a study referenced with a diagram. What we know from that is that if you look at the boys and have a look at when they were diagnosed and how quickly they progressed, and take biopsies from these boys, that the amount of damage within the muscle and the number of CD49 cells within those boys is far in excess of the majority of boys who have had a diagnosis and a more normal progression of what one would expect. So if you look at the boys who are outliers in terms of the way they present, and then take a look at their muscles are so much further damaged, that what we think is that that's probably the outlier that's driving, the outlier in the results when someone doesn't respond. And by the same token, the boy that presents and is diagnosed much later in life and has less damage would be the outlier on someone who would do exceptionally well. So as with any drug that is used is not going to have exactly the same effect on a live organism. When you take a drug and try and influence a biological pathway, each one of us, our biological pathways are different. It will be the same in Duchenne's, but each boy is something slightly different, and the drug is going to react differently in that boy. That's how we explain the outliers. It's based on biology.
Great, thank you, Charmaine. That's all we have time for.
Yep, pleasure.
I'd now like to introduce Dr. Gill Price, who's coming to us from Alabama. He's going to talk about the clinical development plans. Gill, over to you. Your camera's not on. If you'd like to turn that on, please. There you are. Thank you.
Thanks. I'm grateful that we could spend some time with you this evening, and take my part as being able to share with you a few of the highlights on our development plan. I was told by Mark earlier that what you might be interested in is, since maybe you don't know me as well as you might know Charmaine, is to just share a little bit of my background. I'm a physician by training. I've spent the last 30 plus years in the biotech and large pharma world. If my camera was a little bit higher resolution, you could see the gray hair that dovetails with that 30-plus year experience. Mark also did say, though, that rather than that background, that you might be interested here or learn a little bit more about my time at Sarepta. As I'm sure everyone on this call knows, Sarepta is sort of leading the way in Duchenne. In many ways, the reasons why I wanted to come to Antisense were the reasons that brought me to Sarepta initially. Again, you've just heard this compelling presentation by Charmaine about the disease and about the tragedy and the suffering that goes along with the disease. As a physician, I was interested in trying to do something about that. Sarepta was ahead of the curve at that time, and so when I was asked to join their board, I was eager to do so. I do have to say, you're investors, and when I went to the board of Sarepta, we had a similar market cap to Antisense. It was in the millions of dollars. when I left the board at Sarepta, as a result of our eteplirsen success, was in the billions of dollars. And of course, we're hoping to have that same sort of economic success here. So having said that, let's move to the next slide. Talk a little bit about our development plan. So I'm not going to read the slides to you. By the way, the slides will all be available to you on our website, so you can go back to them and review them. What I'd like to do is just offer some of the highlights to each slide. With this, this points out that this will be a multi-centered 12-month two-dose study. We're labeling it as a 2B3. And if that's sort of an adaptive trial, if we get through the 2B part of that, we'll move into larger populations and call this our pivotal study. It'll be followed by an open label extension phase. And again, that's just to collect data over a longer period of time. Very fruitful data in the regard that this is chronic disease, and so we want to make sure that we're having an impact on these boys for a long while, and we believe that that will be the case. In the second bullet point here, there's something that's very important globally, and that is this pediatric investigational plan. We are very fortunate that we have received a final opinion on that, so our PIP was approved, and we were excited about that. The other thing that I would just highlight to you is that it's very rare any longer that a company internally manages the entire process of the clinical trial. The process is typically now outsourced to a contract research organization. In our case, we took a look at several different groups, and based upon our investigation of each of those groups, taking a look at what their experience is, what their abilities and capabilities are in terms of the entire process, forecasting for the number of sites, number of patients, the data collection, the database that will house the data collection, etc. We ended up with what I would describe as one of the global leaders in research, and that's Parexel. I think that was an important decision for us to make. Parexel is renowned in terms of their ability to produce quality data and take that quality data then to regulatory groups that are interested to know that the data is robust and reproducible. The last bullet on here is also a very important point. So who's going to lead the way? Who's going to be your principal investigator? In this case, we were able to secure the efforts of Dr. Thomas Voigt. Again, I'm guessing that for almost everyone on this call, that he is well known. Let's go to the next slide. This cartoon sort of depicts for you what we intend to do. There'll be 3 areas. There'll be a placebo and 2 dosage levels, 25 milligram and 50 milligram. But the point that I would like to highlight on here is the length of this study. So, you know, when Charmaine is talking about pull 2 and these strength exams, we don't expect that to occur overnight in the same way that if you had a sinusitis and you took an antibiotic, you know, you may start to see the effect of that antibiotic. If you're fortunate, you may start to see the effect of that antibiotic in a day, 2 or 3. We believe that this is going to take a bit longer to see the effect. And so we've bit the bullet here to say we need to go out 52 weeks before end of dosing. We're quite convinced that if by taking that amount of time that we'll be able to see an effect, and then that translates into that robust and repeatable data set that I mentioned a bit earlier. Let's go to the next slide. So this cartoon just gives you an idea of timelines. We are working diligently on doing this the right way. And what I would point out, what's the right way? Everybody, it rolls off the tongue nicely. We're going to do it in the right way. Well, the idea is to not try to leapfrog past a particular piece. And so what we've been working on, spending a fair amount of time on, is just the setup. These, as I mentioned, going back to the CRO review. How did we choose the CRO? Well, it was a tremendous amount of work to get us to what we believe will be the best CRO for us. And then now working with them hand in hand to the next steps. Which sites are going to be the most productive? What will the budget be for those sites? When do we actually start or anticipate to have first patient enrolled, and then the last patient visit? And so this timeline just sort of outlines all of that. We're looking at 108 subjects. This is another thing that I would maybe point out to you. 108 subjects for what might ultimately be a pivotal study is not a lot of subjects. I'll give you a comparator. So not long ago, I was named as president and CEO of a publicly traded biotech company here. And we're looking at a study for one of the compounds that we may choose to develop. And we're looking at a 4,000 patient study. So in comparison, our numbers here pale, but it's because we're in this rare space, and it's because we believe that we can show a significant drug effect in that patient population. So is it accelerated? Yes, I think that our program is accelerated, especially when you compare it to something, for example, like a diabetes program. Let's go to the next slide. So what this just kind of points out is the alignment that exists between EMEA and FDA. They're closely aligned. We believe that the best approach to FDA is to take the data that we generate in Europe now, bring that in hand with us to FDA and talk about what that next pivotal study for the United States might look like with this data in hand. Again, knowing that there's close alignment between the two, we believe that going to FDA with this data will really be the thing that pushes us across the goal line. We'll again, when we have that, and we've had one Type C meeting with FDA and we received great feedback from them. But then when we go to them with this for our next meeting with this data in hand, I think what we'll be able to do at that point is really harmonize our clinical development for a global perspective. How do we don't want to, clearly we don't want to be just in Europe or just in North America. We want to be able to take this globally. I think we'll be able to do that. The last thing that I might point out that's not actually mentioned on this slide is we've been working for some time now with the patient advocacy groups in the US. We know that they're very, very important to us. And so what we've been able to establish is a wonderful relationship with them. In fact, they invited me to be on a panel group that is working to develop a guidance for all Duchenne programs. And so I'm able to represent Antisense on that program. And we're working towards the development of that final guidance. In fact, I don't anticipate that will be very far away. I think, Tim, that's my last slide.
That's
and I
right,
might just offer my availability to anyone with questions.
Yeah, there's a couple of questions here. Can you talk to us about kind of dosage? Are there any potential advantages of using a higher dose of 50 milligrams in the phase 2, phase 3 study?
Yeah, that's a great question, Tim. So, dosing, what you really want, you want the lowest effective dose, right? So the idea is with the lowest effective dose, you minimize side effect, right? But you have to, the pivotal word there is effective. So you want to go to the dose that causes or allows for that effect. That's why in this case, we're looking at 2 different doses. And it's not always linear, meaning that the more drug you give doesn't always cause a greater effect. There are therapeutic thresholds that sometimes you can't go through, it doesn't allow for a greater effect. So what I would point out is we're hoping, we're keeping our fingers crossed that the 25 milligram dose gives us a very positive effect. But if we need to bump up, that's why we're taking a look at the 50 milligram dose as well.
And just time for one more, Gill. What do you see as the major factors that could speed up or slow down the clinical development timeline of the drug when both the EU and the US FDA are considered?
Yeah, so again, a good question. The thing that could slow us down is if we don't do these upfront pieces correctly. And I think that that's maybe a part of the reason why Mark was kind to invite Charmaine and I to be a part of the board. Speaking for Charmaine, I would say that we've both done this for a while. In fact, I would tell you and tell the folks on this call, just as an example, I've either led or been a part of 17 successful NDAs. Again, if you had a little higher resolution camera, you might see the gray hair that I have in my head. 17 successful NDAs is a large number. It's thought to be a large number. So what Charmaine and I intend to do is to ask Mark and Nuket and others, we tend to ask them the questions, the performance-related questions that we know should be asked. And that is to do the things correctly up front and don't stumble because we've tried to leapfrog something. So I think with that said, we'll be right on forecast. That's the goal.
Sounds great. Now, we'll just continue now. I think we'll invite Mark back. We're going to talk through the competitive landscape and the market potential. We'll also look at next steps and commercialization considerations. And just please be conscious of time. We've probably got about 15 minutes to go. So I'll let you continue.
Thanks, Tim. Go next slide, thank you. So this slide attempts to sort of capture all the key features of the program that both Charmaine and Gill have spoken to. I guess the upshot is that, we do see this program, this drug as having multi-billion-dollar market potential. It reflects the fact that, the drug is novel in the way that it works. And Charmaine took you through the mechanism of action in targeting CD49D. It is very important to note is that we're the only company in the world that has a drug targeting CD49D in development for Duchenne muscular dystrophy. In the knowledge, again, just repeating what Charmaine said, that we know that Duchenne's boys have elevated levels of CD49D, and as a consequence, it presents as sort of an ideal target for the drug. The other key feature of targeting the drug in this way is that because it works by a novel mechanism, it allows our drug to be able to be used in combination with other therapies. And that's another key feature of the competitive profile of this drug is that we're not contemplating here head-to-head competition. We do believe that the drug will be able to be used in combination, not only with existing therapies that are on the market today, like the corticosteroids or the exon-skipping dystrophin restoring drugs that are marketed by Sarepta, but also those that are coming through development. Charmaine's talked to the exciting preliminary clinical data that we've generated to date, which really sets us up, we think, very well for this last and final stage of clinical development, as Gill outlined. The other key point to highlight, though, is that we are initially testing this drug in the non-ambulant boys, so the boys more advanced with the disease, where there is very little competition, not only that's on the market today, but that's also emerging through clinical development. And at any one time, they're representing at least half the boys with the disease. So, this is a market segment in desperate need for new therapies, and where we're expecting to be one of the few options available for patients. We've got a tremendous IP portfolio that we've established to support the drug's commercialization, and that's backed up by orphan drug designations in the US and Europe, providing additional market exclusivity on top of the patents that we've had both granted and have filed at the moment. Next slide, thanks. So the market today, as I said, key observations are it's very thin, at least the competition as it presents today. You really only have the exon skipping dystrophin restoration drugs that Sarepta are marketing, the generic forms of corticosteroid. In the US only, you have a slightly improved version of the corticosteroid called deflazacort. Interestingly, and important for us as it's the market that we're initially targeting, the only approved drug in Europe for treating Duchenne's, the corticosteroids aren't approved in Europe for this indication, even though they are used there, is this drug Italurin, which is a dystrophin restoring drug, not particularly effective with it in terms of its efficacy, but it is the only drug, and it's only today available for ambulant boys. So it just highlights, again, little competition, and in particular in the ambulant space, we're going into virgin territory. Next slide, thank you. And again, just further to the granularity around the competition, and just really wanting to draw out two key features which I've touched on earlier. There's very little competition happening in the non-ambulant space. We're aware of today one drug only, drug called pamrevlimab, marketed by Fibrogen, that's enrolling non-ambulant patients into a study. And much as I spoke to previously, because this drug works by a different mechanism than Antisense's drug H1102, it looks to address that fibrosis that Charmaine had highlighted was a feature of the muscle inflammation. We're expecting that even if that drug was successful in advancing to the market, we could contemplate our drug being used in combination with it, as we could with the other drugs that are highlighted here. The market's attention has been on the dystrophin replacing agents. Charmaine talked about the gene therapies that are under development by Pfizer, Sarepta. I think Solid Bioscience also have programs. Again, just to highlight, they're limited by the fact that they've predominantly been targeted towards the ambulant boys. We're also seeing them running into both efficacy and safety hurdles. It is unfortunate news for the DMD community, but it does present as being positive news for Antisense and Antisense Therapeutics shareholders, because it does really put the spotlight or focus on H1102 and targeting inflammation as a sensible and potentially a fruitful path forward to treating these patients. Next slide, thank you. We've talked about Sarepta. We're very fortunate to have both Gill and, of course, Bill Goolsbee, the former chairman of Sarepta Therapeutics, guiding us in our development plans for H1102. And of course, that's based on the experience they both gained at their time at Sarepta. Gill talked about Sarepta being the acknowledged leader today, at least in the field of DMD therapy. Their drugs that are on the market today, addressing around about just under 30% of the boys with Duchenne's, because they're targeting a specific genetic mutation associated with the disease. Today, they're selling, well, the last quarter sales for their drugs was nearly 200 million US. So they're on track to be $1 billion selling drug category in the US alone. And I think that's an important point to note, is that the Sarepta drugs actually weren't approved by the European regulatory authorities, because the European regulatory authorities were keen to see data that was showing the drug having an effect on disease progression endpoints, which to date Sarepta haven't actually submitted that data to the regulatory authorities in either the US or Europe. And the reason I highlight that is because that's the path that we're down. And you've heard both Charmaine and Gill talk about targeting pool 2, the benefits that we've shown to date on that disease endpoint, assessing the upper limb function and strength of the boys. It will be the primary endpoint of our phase 2b/3 clinical trial. We've already agreed with the regulatory authorities that would be the primary endpoint for approval of the drug. So we think we've positioned ourselves very well for the European market, but I think our investors should take great heart in the value creation story that is the Sarepta, the fact that that they've been able to take these drugs to market, get premium pricing, but also that we've got the benefit of having both Bill and Gill to guide us on the best way forward to be able to commercialise a drug in this space. Next slide, thank you. So again, time permitting, I won't go too deep on the market beyond the obvious that that the opportunity in Duchenne's, the near-term opportunity is very significant. Markets forecast to grow as big as 10 billion by 2030, and that's sort of reflecting two key features: the high unmet medical need, but also the premium pricing that is being supported for effective Duchenne's medicines. The exon skipping drugs of Sarepta, their average pricing in the US is nearly $1 million annually a treatment on an Aussie dollar basis. And so we see a huge opportunity for our drug, initially targeting the non-ambulant boys, but with the opportunity to expand into the ambulant population. And part of that pediatric investigational plan that Gill referred to, that we've had approval from the European Medicines Agency on, included our plans for the ambulant boys. The other point to note is, and again, Charmaine mentioned our endeavours to expand the application of H1102 beyond the use in Duchenne's into other inflammatory disease settings. We think there's enormous potential there. Anti-inflammatory market space is forecast to grow as 200 billion by 2027. So I think that it's other than the oncology drugs, it's the biggest market segment where we think the H1102 could have a very significant impact. So it should be very exciting for shareholders in the company to contemplate the opportunity in Duchenne's, but also the expansion of the program into these other inflammatory disease settings. Next slide, thanks. Talked about the patent, the portfolio. You see here we've got patents that have been filed that would protect the program out to 2040, potentially extendable for another 5 years, with 7 and 12 years of orphan drug exclusivity available to us in the US and Europe on top of the patent position. Next slide. Next slide, thanks, Tim. Just a couple more to go for me. I know that the market is keen to hear of the news flow pending for the company. The capital raising that we've just undertaken allows us now to advance into the phase 2b/3 program. Gill mentioned that we're with our CRO that we are working on the feasibility evaluations of the 30+ trial sites that we're looking to run in the study in 9 countries in Europe. So that program's underway. Expect to complete that shortly. Next step would be us preparing and submitting our clinical trial applications. They will be done to the 9 countries that we're looking to be able to initiate the study in. The next, obviously, key catalyst will be those approvals coming through. We're expecting to be able to look to initiate the study in those countries, first countries where approvals will come through in the first half towards the middle of next year. We'll be able to report on progress with the FDA, and Gill talked about our endeavours there to be able to get the FDA to assess the data that will be generated from our phase 2b/3 clinical trial in Europe. And of course, another important source of news flow in the short to medium term will be the progress on the new indications, including those initiatives that we're running at the Royal Children's Hospital. One last point to make is that the company has a track record of being able to hit milestones, to be able to do that on budget and on time. And we'd like to think that our success in hitting on those milestones has been major driver of that share price performance that we talked about earlier, taking the market cap from 5 million to 140 million. I'd like to think that should give investors great confidence about the company's ability to continue to deliver on news flow on meeting these key milestone points and driving the share price and valuation of the company further north. Next slide, thank you. Last slide for me, Tim. Not surprisingly, we are being asked by our shareholders about the path forward for the drug, how we're envisaging the commercialization of this program. And I guess at a high level, the view would be, we think we've got optionality when it comes to the way forward with the program. What we want to say is that now that we've raised capital, we are laser focused at moving the program into this phase 2b/3 clinical trial. And we are setting this up as a registration study. But what we are also thinking about is, beyond gearing ourselves up to be able to take the program all the way through to an approval in Europe, and that having Gill and Charmaine, with us to be able to anticipate that the requirements of the company, including increasing staffing levels and looking to strengthen the board further from what we've already done today. We're also starting to think about the prospect of being able to take the program to market ourselves, and Gill touched on that was what Sarepta were able to achieve in the US. And the reason for contemplating that is a simple one. If we're able to get approval to take the program to market, the value creation is undeniable. And it takes us from R&D company to a vertically integrated pharmaceutical company with sales, and companies get richly rewarded for doing that. The other important point that may not be known or evident to our shareholders is that, and it's one of the advantages of working in the rare disease field, is that the number of treatment centres and the number of physicians treating Duchenne's is relatively small. You can see here in the 5 major markets in Europe, it's estimated to be around 180 treatment sites and only around that 800 physicians, with a handful of key opinion leaders who are influencing the prescribing practices. That would compare to 20,000 GPs here in Victoria, Tim. So it allows a small company like Antisense Therapeutics to contemplate a focused marketing and sales effort to be able to target that sort of population. And so we think it's achievable. We're also we are incentivized to take this program into this advanced stage of development and onto the market through the arrangement we have with Ionis. Simply put, we take the program to market, we get a much greater percentage of the benefit than if we were to partner it. But we also contemplating perhaps the opportunity to partner the program on a geographical or indication basis. That also presents to us. So I guess what we say is that we're looking at the options. We think we're really well set with the experience that we have around the board to direct us on the path forward. And I think investors should be excited about the prospects here for the company and about what can be achieved. And obviously it would put us in very, very rare company if we could take this program all the way through, and we think that is achievable. Thanks, Tim.
Thanks, Mark. Gill, we might ask you online as well. You might want to turn on your camera if you don't mind. Thank you. There's just a couple of specific questions around the US. Now, you've obviously had, you were very successful. You served for 9 years or so on the board of Sarepta Therapeutics, which was the company to first introduce a drug approved for Duchenne's. What's the US market look like in terms of your contacts, your experience? How can you help Antisense Therapeutics open up that US market?
Yeah, so a couple of things. One, I would just go back to to what Mark sort of outlined in terms of the number of physicians that are treating the disease, the number of specialty treatment areas, etc. We actually had very, very similar discussions at Sarepta. I mean, remarkably similar discussions at Sarepta and then at the end of that, we convinced ourselves that we could take the drug through commercialization. Again, just kind of going back to that comparator that I shared with you earlier, you don't think that a company of our size, of this market cap size, could take a drug for diabetes, for example, a GLP-1 inhibitor, and commercialize that. That program requires an Eli Lilly, for example, with a couple thousand sales representatives, et cetera. This is just very, very different. And so we had those exact conversations, and we're having them again here with Antisense, and I think we'll come to the same conclusions. To get specifically to your point, so JP Morgan is coming up in early January. I've discussed with Mark, I'm on the board, so I'm happy to be a representative to anybody who has a question about Antisense at JP Morgan. So we'll have some presence there. I think ultimately, Tim, it's going to be important to bring the drug and to bring the studies to the US so that we can start that process of introducing this compound into the US and into the Duchenne environment here. And so we'll get to that. That's something that we do, as Mark sort of hinted to, as we get through this European piece, as we begin to scale up a bit, we'll come to the US. And clearly, in terms of the key opinion leaders here, I'm familiar with them all. I have several of them on my phone now that I can call up pretty quickly. So I think we'll be in very good shape. Also, by the way, again, Parexel has a tremendous presence here, so that we won't have to go through the troubles or the difficulties of trying to migrate things from one CRO to another. Hopefully, we'll be able to continue on with Parexel here. And so between the key opinion leaders that they know and the ones that I know, I think that we'll have a seamless transition into the US.
And Mark, I might just get you to unmute and just ask you finally just the last question, and we might wrap up there. So how do you maximize value for shareholders? Do you actually take this product to market, succumb to a buyout, or do you partner? If you do partner, when do you think would be the opportune time?
Yeah, sure, Tim, and that was, if we go to the previous slide, was what we were endeavouring to address here, and we'll speak more to this, Tim, in time. We are very conscious of that value creation story at Sarepta, and and having Bill and Gill there to guide us does give us a lot of confidence that could be a very a fruitful way forward for the company, and achievable one. Because so the other aspect of course is that we need to be mindful of what partnering interest may emerge. The board is aware that that we may get offers from partners that are attractive, and so that would be contemplated, of course, in these decisions that we'd be making around moving forward and when to partner. I mean, we raised money and we're down the path of raising additional funds with the goal of moving into this open label, this phase 2b/3 clinical trial. So that is the focus of Tim, and so that is really where we're headed with the program in Europe. We have said the company may contemplate partnering on a geographical and/or indication basis, but I think that we'll be able to speak to that more in due course, and so we really wanted to open up the dialogue on it, Tim. This is the first occasion that we've spoken on it. I expect we'll have more to say at the AGM on it. I'd invite Gill or Charmaine to add to that, but we appreciate and understand why shareholders would be asking the question. But last point on is that I think we've just positioned ourselves perfectly with having the experience on the board to be able to make these calls.
And Gill, any last words from you before we wrap up this afternoon?
No, again, just grateful to be here, and so thrilled to be a part of the Antisense story. I mean, I feel like we hit a home run at Sarepta. And you look back on your career, and if you hit a few singles, one or two doubles and a triple, you feel good about it. If you have a home run, that's wonderful. I'm blessed trust me when I say I'm blessed to have a chance to hit a second home run here at Antisense, and I'm eager to do that.
Great, Gill, and great, Mark. And thanks for your time. Great presentation, very informative. Always a pleasure to have directors on board, and thank you, Charmaine. And we'll finish up on that note. Good afternoon.
Thanks very much, Tim.